this is Jawad Ahmed from Medico's Lectures by JD, and in this video we will be covering the parasites of the class Telosporea. So first of all we will be covering Plasmodium and the disease caused by it, that is the malaria, in full detail, and we will also be covering the Toxoplasma and Cryptosporidium hominis. So these are the parasites that we will be studying in this class. So let's have a start. Before going directly to this class, first of all, like always, a brief overlook over the previous videos, that what we have studied so far. So first of all, we have divided the parasites into two groups. The one is the protozoa and the other is the metazoa. So this classification is on the basis of cellularity. So if the parasite is unicellular, we have placed that into the protozoa, and if it is multicellular, we have placed this into the metazoa. Now the protozoa again are then classified on the basis of the organelle of locomotion into the class Rhizopoda, and they have pseudopodia as their organelle of the locomotion. Second, the class Zoomastigophora have flagella. The third, Ciliata, they have cilia, and finally the Telospora that we will be covering in this video that don't have any organelle of locomotion. So they don't have any organelle of locomotion. Then if we come to the class Metazoa, then we have divided into the platyhelminthes or the flatworm, and also the nematohelminthes or the roundworm. So this classification is on the basis of the body structure. If it is flattened, then we have placed them into the platyhelminthes, and if they are rounded, we have placed them into the nematohelminthes. Then the platyhelminthes again is classified into the class Cystoda. They are also called as the tapeworm. Their bodies are segmented, and each segment is broader than longer. So this is again classified on the basis of the body shape. And the second one is the class Trematoda. So the Trematoda are the flukes. They have a leaf-like body. So this is the general classification of the parasites. So we have covered the class Rhizopoda, Zoomastigophora and Ciliata into the previous videos, and today we will be covering the parasites of the class Telosporea. So let's have a start. So before going to the parasite, just a brief overlook over this class properties that will be mainly present in each of the parasite that we will be studying. So first of all is that they don't have any organelle of locomotion, as we have said. So just have a look over here. This is a Plasmodium. As you can see, it doesn't have any cilia, flagella or pseudopodia, so which are the organelles of locomotion. So it doesn't have any one of them. Second thing is that these are the intracellular parasite, means they will cause the disease on the cellular level. They will enter into the cell and they will cause the lysis of the cell. As you will see that in this case it will cause the lysis of the hepatocytes and the erythrocyte in the case of the Plasmodium. So these are the intracellular parasite. And the third property is that is very much important, that they have a very complex life cycle, in which they have a sexual life cycle and an asexual life cycle, and both of these life cycle will alternate with one another. So those they have alternation of generation between the sexual and asexual reproduction, and their asexual reproduction is called as schizogony or schizogonic cycle, whereas the sexual reproduction is called as sporogony or sporogonic cycle. So you have to remember these terminologies. The asexual life cycle is called as schizogony, whereas the sexual life cycle is called as sporogony. So these are the definite points that you will see in each of the parasite that we will study in this class. So keep these in mind, that they don't have any organelle of locomotion. They are intracellular parasite, and they will have an alternative sexual and asexual reproduction, and this very thing make them very complex to study, because they have various different morphological form in this way that we will study, as you will see. So let's talk about the first of the parasite that we will be covering is our Plasmodium. So Plasmodium is actually not the name of the parasite, it is actually a genus name. You can see over here. So this is a genus, and these are the this genus include those species, those parasites that cause the disease called as malaria. So there are 4 parasites that we will cover in this genus that causes malaria in man. So these 4 are, first of all, the Plasmodium vivax, which cause benign tertian malaria, the Plasmodium malariae, the Plasmodium ovale, and the Plasmodium falciparum. So these 4 are our parasites in this genus. So actually we will cover these 4 parasites in this video. So these all these parasites are causing malaria into the man. So Vivax is causing benign tertian malaria. The malaria, Plasmodium malaria is causing quartan malaria. The Plasmodium ovale is causing benign tertian malaria, and Plasmodium falciparum causes malignant tertian malaria. So the difference between the tertian malaria and the quartan malaria is that in the tertian malaria, the fever that you will see that in malaria there is an intermittent fever. That is, there will be a period of fever and then there will be a period of relief. So this in the tertian malaria, the fever will occur after every 1 day. That is, at the alternative days the fever will be present, whereas in the quartan malaria the fever will be present on every second day. So this is the difference between these two. And the benign and the malignant, so the malignant is used with the Plasmodium falciparum because it has high mortality rate. Due to that it causes severe complications, and due to that its mortality rate is high. That's why it's called as malignant tertian malaria. So now we will be studying and we will be covering these four parasites at one time. So get ready to view a large number of the morphological form, because we are studying four parasites at one time. So now let's have a start at its morphology. Now you know that these organisms, they don't exist as one form. Say for example, I am Jawad, I am just in this form, but no, these organisms exist in various different form in their life cycle. So just a brief overlook over their life cycle. I will first give you just a brief overlook over their life cycle so that you can remember these morphological form. We will study their definite life cycle again later. We will study shortly, means after this morphology we will study their life cycle, but just a short overlook over their life cycle so that you can remember its morphology. So first of all, we have studied that they have this property that the sexual cycle and the asexual cycle alternate with one another. So first of all, in its asexual cycle, which occur in man. So in man, the asexual life cycle of this parasite take place, and that's why the men are called as its intermediate host. The intermediate hosts are those hosts in which the asexual cycle of any parasite take place, whereas the host in which the sexual life cycle of the parasite take place, we call that host as definitive host. So men are its since in men asexual life cycle take place, so men are its intermediate host. On the other hand, the mosquito, in which you will say that the sexual life cycle of this parasite take place, so the mosquitoes are its definitive host. So first of all, in its asexual cycle, that is in men, then when a mosquito infect a man, so first of all that sporozoite, the mosquito injects the sporozoite into the man. So that sporozoite will first enter into the hepatocyte, causing complication over there. But just remember that when the parasite will enter into the red blood cell, so first of all you will see the early trophozoite form. The early trophozoite form means first of all the plasmodium will arrange itself in a ring form, so that we call that form as its early trophozoite form. Then it will convert into a mature trophozoite form. Then a schizont will be formed. After a schizont, a large number of merozytes will be released. The schizont will burst and the merozyte will be released into the blood, and some of these merozytes will again enter into the liver, and then they will cause a relapse infection, and those that enter into the liver and cause relapse infection, those are called as hepnozyte. Whereas the other merozytes, some will cause again lysis of the other RBCs and the infection, and will continue the infection, whereas some of the merozytes will convert into the gametes and will start the sexual life cycle. So the sexual life cycle will have a macrogamete or a female gamete and a microgamete or male gamete. Then after the fusion, zygote will be formed, and through various different steps, then after that a sporozoite will be formed, and this sexual cycle will be occurring in mosquito, and after that the mosquito will inject the sporozoite into human, and then the cycle will repeat. We will study this life cycle in detail, but just to remember the different morphological form, I just explained it a little bit earlier over here. So you have to remember the different. Now we will study the different morphological form, of course, the different their shapes and their everything. So just remember that these are the different morphological form that we will study. So, and among these, the important one are this early trophozoite form, the mature trophozoite form, the schizon, and after that the true gametes. So you have to remember these because these are important for diagnostic purposes. So let's have a start. So first of all, the early trophozoite. We studied that first of all, early trophozoite will be formed, the ring form will be formed into the RBC. So in its ring form, you see that in every one of these, just you see that this is your plasmodium. This plasmodium has arranged itself into in a form of ring. You can see this whole is this is the red blood cell RBC. So inside the RBC, you can see over there this ring inside the RBC is the plasmodium. Now in this plasmodium, this is this bluish, as you can see, this is the cytoplasm of the plasmodium, whereas this red dot is the nucleus. This is the nucleus of the plasmodium, and in the center is a transparent area, this vacuole like looking area. This transparent area is it is a transparent area of the plasmodium. So this is the plasmodium, a bluish cytoplasm and a nucleus and a central transparent area. You can see over here bluish cytoplasm, nucleus and transparent area in each of these. Now how will you differentiate that which of these four parasites that we have studied, the Vivax, falciparum, malaria or ovale, is causing the malaria? So inside the RBCs, first of all, in Vivax you will see, in Vivax, malaria and ovale, you will see a thick ring. You can see a thick ring over here, a thick ring and a nucleus, a thick ring and a nucleus. You can see in all of these three, you will see a thick ring and a nucleus, whereas in the Plasmodium falciparum, there will be a very fine ring. You can see a fine ring over here. So in case of Plasmodium falciparum, this ring will be fine, whereas in case of the other three, this ring will be thick. And the other thing is that in the Vivax, malaria and ovale, this ring will be 1/3 of the diameter of the RBCs. This ring is 1/3 of the 1/3 of the diameter of the RBCs, whereas in the Plasmodium falciparum it is smaller and it measures about 1/5 of the diameter of the RBCs. So it is a smaller ring. It measures about 1/5 of the um, diameter of the RBC. The other thing about falciparum is that you can see two rings in one RBC. You may see two rings in one RBC, and this will show us that multiple infection, that this RBC is multiply infected. So this is your trophozoite form, and this is how you will see them in the blood smear. Now let's move forward to the mature trophozoite. First of all, I have told you that first of all, when the RBC is infected, so first of all early trophozoite will form. That early trophozoite will then convert into the mature trophozoite. Now what will happen in mature trophozoite? As you can see, first of all in Vivax, so in Vivax it will be irregular in form. You can see that it looks like different pseudopodias are formed. So this is irregular form. So in the Plasmodium Vivax, the ring will be irregular in form, whereas in the Plasmodium malaria you can see a band, that the organism have arranged itself in the form of band. On the other hand, in the case of the Plasmodium ovale, it is rounded and compact, and there is no band formation, or there is no irregularity. As you can see, it is a proper ring. On the other hand, in case of falciparum, the large trophozoite form is not seen in the peripheral blood, because it then leaves the peripheral blood vessel and it enters into the blood vessels of the internal organs. So the peripheral blood doesn't have doesn't mainly show the Plasmodium falciparum. But you have to remember these three, that the peripheral blood will show, in case of Plasmodium vivax, this kind of irregular ring; in case of Plasmodium malaria, a band; whereas in case of the Plasmodium ovale, a definite ring. So that you have to remember. Now move forward, that this trophozoite will then form a schizon. Now what exactly is a schizon? So you can see that the nucleus of that we see, the large trophozoite, that will divide. This is early schizon and this is the mature schizon. The second row is showing the mature schizon, whereas the upper row is showing the developing schizon. So first of all, the nucleus divide, and you can see the different fragments of the nucleus, and then it is surrounded by the cytoplasm. We have studied the bluish cytoplasm of the Plasmodium. So this nucleus is then surrounded by the cytoplasm, and when this cytoplasm surrounds this nucleus, After that, what happens? So these are your various type of schizon. Now you have to remember that the difference in between them, that first of all, the schizont of the Plasmodium vivax, you can see it's irregularly arranged cell, and there will be irregularly arranged, and the inner cells, these I am referring them to a cell, these are actually merozytes. So there are about 12 to 18 merozytes inside the schizont of the Plasmodium vivax, whereas in case of Plasmodium falciparum, these are large in number, so these are mainly 24 merozytes. So in case of Plasmodium vivax, these are 12 to 18. In case of Plasmodium falciparum, these are 24 in number, and these are irregularly arranged. On the other hand, in case of Plasmodium malariae, they are arranged in the form of rosette. You can see these are very beautifully arranged into the form of rosette. And in case of Plasmodium ovale, again irregularly formed cell, and these are mainly 8 merocytes that are irregularly arranged. So 8 merocytes in case of Plasmodium ovale, 24 in case of Plasmodium falciparum, and 18 and 12 to 18 in case of Plasmodium vivax. Whereas in case of malaria, these will be arranged in the form of rosette. So you have to remember all these forms. Now, okay, so now we will talk about the gametocytes of the Plasmodium. So we have talked that the schizons liberates a large number of merozytes. Now some of these merozytes will then again causes the lysis of the red blood cell, whereas some of these merozytes, they develop into the micro and macro gametocytes. Now the macro gametocytes and the micro gametocytes. So let's have a look at the different macro and micro gametocytes, as you can see over here. So how will you differentiate between the different types of the different species of the Plasmodium? So first of all, in the Plasmodium vivax, malariae, and Plasmodium ovale, the Plasmodium will be the micro or the macro gametocyte. The gametocyte will be circular in shape, as you can see here. So it will be circular in shape, and the nuclear content will be intact. Whereas in case of the Plasmodium falciparum, the Plasmodium is crescentic in shape, as you can see here and here, and moreover the nuclear materials is dispersed or scattered. Now in the case of Plasmodium falciparum, it is very diffuse, as you can see, and it gives this Plasmodium a reddish look. Now as you can see, the Plasmodium vivax, malariae, and Plasmodium ovale, they are all circular in shape or rounded in shape. Now what is the difference between these three? So first of all, the Plasmodium in the Plasmodium vivax, the RBC will be enlarged. In case of the Plasmodium vivax, the RBC will be enlarged, and as well as in the Plasmodium ovale, the RBC will be enlarged. So in these two, the RBC will be enlarged, whereas in case of the malaria, the RBC will either be smaller in size or it will be unchanged in size. So in this way, you can differentiate between them. Moreover, you can see that you will see that the Plasmodium ovale, the Plasmodium ovale appear early in the infection, whereas the Plasmodium vivax appears late in the infection. So in this case, you can differentiate all these gametocytes. So that was all about the gametocyte. Let's give it a very quick a brief overlook. So first of all, we have studied the early trophocyte, the ring form. We have seen that the malaria and ovale has a thick ring, whereas falciparum has a small ring. Then we have studied the mature trophocyte stage. You get irregular shape, it has got a band and this that. Then we have studied the developing schizon, and finally which develop into schizon, the nucleus, you can see over here, it divides, and then the cytoplasm gets around it, and the different arrangement of the different Plasmodium. So as a result, schizont it forms. Schizont liberates then merozyte. These merozytes are then released, and these merozytes then some of these merozytes form the micro and micro and macro gametocytes. So once these gametocytes are formed, we have discussed that the Plasmodium vivax and malaria as well as ovale, these are rounded, whereas Plasmodium falciparum is crescentic in shape. Plasmodium ovale can be seen early during the infection, whereas it is seen late in the infection. And moreover, Plasmodium or malaria, the size of the RBC is either decreased or it remains and normal, whereas in case of vivax and ovale, the size of the RBC is enlarged. So these are all the morphological form that you should remember, and that you should just memorize these form, because these are diagnostic, and it is very important to know these, because in the blood smear you will have to differentiate that which of the Plasmodium species is causing the malaria, and then you have to treat the malaria accordingly. So I hope you have gone through all these forms. Just memorize them and just remember the differentiating points between them. Now we'll come towards